So if it doesn't have eyes, how does it navigate?
It uses lots of other senses, like its sense of smell, and its sense of touch. It can feel vibrations and navigate this world that is 100% of the time in the dark, using many other senses. Us as very visual creatures, we forget that there are other ways to feel and experience the world outside of our vision.
Another one of these large black corals that we got a good zoom in earlier.
Oreo.
I think when Jake starts identifying all the organisms, it's time for Megan to start flying Hercules.
Yeah, let's do it. Switch seats.
I'll navigate.
Yeah, I'll fly.
I've done a bit of navigating in my day.
I've done a bit of flying in my day.
Perfect. All right.
We're a match. Yep. Oh, there's a bolosoma off to the right.
Yeah, we hadn't yet.
The white sponge.
Yeah, not documented this one.
Down low?
On my...
Yeah.
Oh, that one, okay.
Oh yeah, I guess there's a lot of white sponges. Stalked sponge.
Can push in there.
So this is a glass sponge in the family Euplectellidae called bolosoma. And it has a little friend, a little hermit crab hanging out on the top of the sponge.
Nice.
Thanks, Jake. That's
There was
great.
a cool shell on that yellow coral next door too. That's gonna make my highlights stills. It's a good shot.
Calendar. Okay, come on.
Oh, we have a question. It's from Annie and her students. So what does Libu's voyage mean, and how does that tie into what you guys are doing?
This voyage was named after Prince Libu, who was the son of High Chief Ibadul of Koror, and he was the first Palauan to go out and explore with some British people, I believe. These British people, they were exploring, and their ship
ran aground.
Yes, exactly. Thank you. It burned ground in Oolong, and so High Chief Ibadul and his people helped them repair the ship, and in return, they took Libu and took him to England, I believe, and to further his studies and explore the outside world. But unfortunately, Prince Libu got smallpox and could not make it back home. So this voyage is in honor of Prince Libu.
Yeah, and a couple more things here to add. So when we started planning this expedition, this voyage, a process that really started about 2 years ago, really right after we knew about this commitment and the possibility of ships coming to support ocean mapping in the Palau National Marine Sanctuary, we started engaging various stakeholders and how we can best use these expeditions to address local priorities, what kind of information we could gather, what kind of activities could we include in this expedition to support the priorities. And early on, we worked with a wide group, and this was facilitated by the Palau International Coral Reef Center, and also included One Reef and other partners with traditional knowledge, and following an endorsement by the Council of Chiefs and the Society of Historians and Coral House of Traditional Leaders. They began the process for developing a name for these expeditions. And so after rigorous consultations and considering various factors, they settled on this name. And several other activities also part of this mission to honor this and weave in traditional knowledge and cultural practices into these expeditions. And we're very grateful to our partners of the Palau International Coral Reef Center who have been part of this process from the get-go. And it's very exciting to, on this expedition, include various activities. So culture activities prior to sailing, several of us were very fortunate to be part of a cultural briefing. For many of us that we don't call Palau home, but we're very fortunate visitors, so we got a really nice orientation. And also modern science being woven into this. So the Palau International Coral Reef Center has been working for a while to start characterizing Palau National Marine Sanctuary. And maybe Maikani, you can tell us a little bit about the eDNA sampling program.
Fish up to the left of lasers.
So Pickrick started this eDNA monitoring program back in 2021. Where we go out and we sample a bunch of different sites within the Palau National Marine Sanctuary, and we extract the DNA that we get from those samples, and that way it helps us determine what organisms live within our waters. Since 2021, we have gone out and sampled about 4 times. And those samples we bring back to Pickrick, and we send them off to our collaborators at Stanford for further analysis. And
Yeah, thanks for that, Maikani. And Maikani and Piera have been, in addition to standing watches here in the control van, they've been working very long hours in the wet lab, processing eDNA samples collected both with the remotely operated vehicles and from the surface with a hand-launched Niskin bottle, and collected a whole bunch of samples over this expedition, and that will continue also on the next voyage, on Lebu's Voyage 2, which is set to depart on Monday.
Yeah, a little heterocarpus shrimp. There's a toppled over Victor Gorgia, that purple coral. Some more of that period glass sponge that was super abundant on yesterday's dive. But here, this seamount seems to be more towards the corals. We're seeing a high diversity of corals, a lot of these acanthic gorget corals, as well as some unique sponge species.
Yeah, that furriate sponge was a really nice collection. It actually remained quite intact.
Oh, fantastic.
And was beautiful to look at in the lab. There was several associates living inside of it as well.
Can we get a quick zoom on the...
you to, yeah, push in there.
So this is a hard coral called Analopsamia rostrata. And that's what's making all this coral rubble that we're seeing everywhere. So there's got to be more of it somewhere, you know?
That's great. Thanks, Jake.
Okay. Come on.
Bridge, nav. Can we make a 30-meter move 068? Fantastic.
So a question about Hercules and Atalanta. How long is the tether between the two ROVs?
About 30 meters.
Okay.
And that's also a neutrally buoyant tether. And that basically allows Hercules to be completely decoupled from the motion of the sea surface. And that's why you get that stunning imagery, completely still.
There's 2 floats and a weight on the tether to help manage the shape of it as Atalanta's above Hercules.
We can take a look at that anemone. So this big anemone is in the family Actinostolidae.
We can go for a zoom there.
I think they're really pretty.
Yeah, definitely.
I also really enjoy the little bulbs on the ends of the tentacles. That's fantastic, thank you.
Okay.
What's the spiky thing?
Spiky thing.
It's off to the right.
Ooh.
Oh, it's another different type of anemone. This anemone is in
Push in there.
the family Actiniidae, I believe.
Okay.
Cool, thank you. Oh, there's a nice big heterocarpus shrimp in that rock.
Yep.
We did see a lot of them on the baited camera deployments. Oh, there's a tiny crab. It might be similar to the one we were trying to catch, but this one looks like it might be smaller. So more suctionable.
Push in there. Can go for a zoom there.
Yeah, that's the same one, but smaller.
Okay.
You think we want to try for it?
Yeah, if Jake feels like
Yep.
this is a good spot.
Yep. Yeah. Yeah, let's do a power of that. Yeah, tilt the camera down. Jar number 3.
Ugh. Oh.
Oh. Almost.
Well, it was a good attempt.
Yeah.
Yeah, if it gets too difficult, I mean,
Those crabs.
yeah,
They're fast.
that's all right.
I was just like, it's smaller. Maybe it'll be easier.
So we have a question. Does do the fish that live in these depths live alone, or are there any particular species that live in schools?
Most of the fish down here do not school, so they live solitary lives.
Yeah, the only time really where you see big aggregations of fish in the deep sea is typically when they're all feeding on the same thing, either a whale fall or something that's there, and a lot of the scavengers will all congregate around them. But yeah, you do
pushing on
not
this
have the
fish.
schooling behavior that you have in a lot of in the sunlit ocean and shallower depths.
This one's cute. Looks like monometopus, a type of cuskeel.
Oh. All right.
And a lot of these questions about comparing shallow and deep ecosystems, if you look at the root cause of a lot of those things, they have to do with food availability and the stability of these environments. So the deep sea is typically, there's a lot less food than you have And so things are more, they're slower growing, longer lived, reproduce later in life. And then you also tend to have, you don't have the seasonality you have. Now this is kind of permanently dark, permanently cold. You don't have summers or winters. You do have some seasonality in the form that, if there's big things that, where you have blooms at the surface, and then you have periods where there's food raining down and periods where it doesn't rain down. But for most cases, now these are very stable environments, the deep sea. It's been the same kind of temperature, the same darkness for thousands, if not millions of years, and so these species are also not adapted to change. They're very different than your weeds in the garden, where you would cut the weeds and then a week later they would be back. Now these things, they are not adapted for changing environments, so when you perturb these ecosystems and change these ecosystems, the species, they're just not equipped to basically bounce back and come back. And that's really the importance of places like the Palawan National Marine Sanctuary that protect a lot of these deep water ecosystems, because these are highly vulnerable ecosystems. If you were to perturb these habitats, it would take very, very long for them to recover. And in the deep sea, there have been examples where trawl marks that were left hundreds of years ago, and that still look very similar today than they look back then. It just doesn't turn over. These are relatively stable environments.
So Daniel, we're up here on the ridge, and we're seeing a lot more coral, but we're also seeing a lot, just naturally, some dead coral and possibly some fossilized coral. Is there any reason why we're not taking samples of those or having interest in those?
Yeah, that's a very good question. So I mean, the coral, all those white, the reef building corals, that's a pretty well studied species. There's about 6 reef building species in the deep sea, and they have been pretty well studied. I think we have a pretty good sense of what they are, so there wouldn't be something about. And then in terms of collecting these samples to try to get more of a geological insight, I think for a rock, volcanic rock would be a bit more advantageous. Yeah, when we started this process of planning for these expeditions, we put out a call for science input, and that basically allows the broader science community to just say what they think in the places that we're working in, what are the important places we should study, what are the type of features we should study, what are the types of samples we could collect to advance theories or studies that are happening there. And we haven't heard anything about that particular species and that particular, and so yeah, we try to basically, when we design these studies, is to try to use the samples in a way that it helps as many scientists as possible. Yeah, and that's why basically we're trying to basically focus on some things and not others. And yeah, these types of rubbly environments, Megan pointed this earlier in the dive, is when you have these deep water coral reefs, they're quite a bit different than shallow water reefs. They're not as cemented together as what you have in the shallow waters. So you basically have these big white corals that will then topple over, and then you have a new coral that grows on top of it, and then you basically rinse and repeat that thousands and thousands of times, and you'll start building mounds, and over time we'll build bigger reefs. So it's a slightly different kind of shape of these reefs than what you have in the shallow water environments. So there is active research in some parts about trying to understand the growth rates of these reefs. But that's typically in places
Can
where
we
you have,
see this middle yellow one that's like slightly yellower color, not the golden ones?
yeah, where you have more of a time series where people have gone
That has
back
the
to the
snake
same
star on it.
reef over
Yeah,
time.
with the
And
lasers just to the right.
this is for the very first time that anyone has ever been at this place and has ever
All right, Aaron, push in.
surveyed it.
Yeah, this one looks different than the other ones.
So I hope that addresses your question.
Yeah, thanks. Thanks.
All right.
Yeah, this is a different coral than what we've been seeing. We've been seeing this Acanthogorgia, which is that lighter yellow on the top there, and we've been seeing those more goldenly ones, but
Yeah, come
this
down.
brighter yellow one is new. So it's confusing because we've got like 4 different yellow corals all together. What do you think, Daniel?
Do you want me to zoom back in?
Yeah,
Yeah,
let's.
let's zoom
you can
back
zoom
in.
back in.
Yeah, I think we have some convergence here that this could be a.
I just wanted to give you a chance to settle.
Yeah,
Thanks.
appreciate it. Yeah, I think if we're set up.
All right. Bridge, nav.
Yeah, Sako is chiming in here as well.
Can we hold position?
Yeah, that's very different than what we looked at yesterday.
So we're sampling this? And
Yes.
Yep. Okay.
Yeah.
I thought I heard that. I just wanted to make sure. Okay, and how, I mean, is this whole thing going to come off?
I think it's just going to pop off the rock.
Okay. So if I try to get it low?
Yeah, low is probably better.
Do you want a little zoom in now?
A little zoom would be
Okay,
good, yeah.
cool.
Just gonna crush there, but yeah. Okay, starboard box.
Yep. If you can get it in one of the little ones, otherwise.
I think so. Yeah, that's
Okay.
about the right size. Okay, switch over. Bump out the box. Snaggletooth.
And Asako thinks we saw this a little bit earlier in the dive as well. So yeah, great collection.
We go.
Nice. A little bit of a tight fit, but well done.
Wonderful. Megan, can we get just a estimate of range to waypoint 3, please?
Of course. It looks like we've got about 300 meters.
Roger that. All right, well, we get situated here to get underway. Just also want to take a moment to thank our very loyal and relentless scientists ashore, joining us at all hours throughout the day, throughout the night. We've had a really great and dedicated group of scientists joining us. Aaron Judah from the University of Hawaii, Asako Matsumoto from the Chiba Institute of Technology in Japan, Chris Kelly from the University of Hawaii, Ike Oto, Palau International Coral Reef Center, Jonathan Womack, independent historian Kenneth Sulak, retired from the U.S. Geological Survey. And I should also acknowledge our scientists on ship that whether they're on watch or not, they're also watching. So I know Brian, Jeff, Harold, and Pierre are all connected right now. Whether they're on watch or not, it's a bit addicting when you start doing these things, and just so much cool things to see. And I think people are going to have withdrawals here over the next couple of days when we're not diving.
I see we also have some science communication fellows tuning in. They will be participating in next week's voyage and are saying they're very excited to come to Palau and kick off next week.
Yeah, we're so excited. Yeah, we have had really fantastic science communication fellows on this first voyage, Lebu's voyage. It's been a privilege to work with Imade and share watches. Also Lottie and Elle, a wonderful team helping us connect the work that we're doing here with schools throughout Palau and the Pacific Island region. Thank you so much for all the hard work.
Do we know how many they've done? Because they're constantly in there.
Megan usually keeps a tally.
Yeah.
Megan, at the end.
It's impressive.
It is. It's non-stop.
So we have two questions that just came up in the chat. One is asking if there's been anything that's been hostile towards the ROV.
We get that question a lot. But,
Yeah.
sometimes we get crabs with their claws up like this. And it looks like they would want to be hostile, but I think it's just defensive.
Yeah, that's sort of just what they do. Well, we have had a swordfish kind of be territorial towards the ROV. They're very territorial animals.
And the second thing, the viewer says the ONC expeditions do a lot of research with cameras and other equipment. And so has there been something similar in the protected areas around Hawaii?
So ONC, like if you're talking about the Deep Sea Observatory, is the largest one in the world, has many nodes and over 500 CTDs and other instruments. In Hawaii, we have the deepest deep sea observatory in the world at a depth of 4,728 meters. And it only has a couple nodes and a few instruments, and the University of Hawaii helps maintain that
Push in there.
deep sea observatory and monitors all the data that it collects. There are ocean observatories all around the world, and I believe there are more being built even now.
Yeah, looks like a Kumba. I think Jeff is wondering if we're positioned to do so, to try to attempt a collection.
You come down a bit, 5 meters or something.
And this is the one that got away from us yesterday.
Okay, and you can come wide on the video. Yep, that sounds good.
For these mobile animals, Jake, would it be easier if you only had one job?
Probably.
We could probably make that happen if it would be easier.
Let me grab it.
Oh,
Stuffed it. That's very soft. Let's swim right into it. Oh, well,
It's something.
interesting tactic.
That's one way to not get caught.
I should have opened the box.
Yeah. Yeah, I think this might be a
Yeah.
tricky one. And
yeah, these fish are
yeah.
difficult.
Oh,
Suction's on.
Oh,
Oh, now it's underneath.
it's too fast.
Yep. All right.
Yeah, it was a good try. Well, thanks, Jake. And we'll get situated here and continue moving forward. Looks like we've covered a little bit of over a third of the way between waypoint 2 and 3. And we'll continue moving along this ridge. Waypoint 3 is about 1 kilometer from where we started. One more thing that might be good for, at a very basic level, going back to the question, Imade, you posed about the observatories. Now, observatories are a fundamentally different way of deep sea exploration. Now, those are set up to basically go to the same area and collect information at different points in time and try to understand how things change over time or if they're stable. What we're doing here is basically going to a single point at a single point of time and then looking what's there and then going to a different place. And so yeah, you get very different information with these types of explorations. Now we get snapshots of what the environment is at this point in time. We don't know if we were to come back here tomorrow. Obviously the corals, the things that grow attached to the rock, they would still be there, but some of the fishes might move away. And yeah, so observatories are really powerful ways of studying things that change them on different time scales.
Is this the one we saw earlier?
Yeah, I think it's the same as the one we saw earlier.
I think so.
So, question for Jake. How hard is it to collect fish using the slurp versus using the net?
I haven't tried using the net, but the slurp is definitely hard, especially when the fish are fast and they're not sitting there on the bottom. You have to get really close and try not to disturb them before slurping them, and then it also depends on the size of the fish too. The bigger the fish, the harder it is to get into that small nozzle.
Yeah, also the shallower we are, the fish tend to be slightly better swimmers. Bridge, nav. Can we make a 40-meter move, 068? Thank you.
Also just here while we're moving along the ridge, a little bit of situational awareness, particularly for our viewers that are not from Palau. So our closest point of land is about 30 miles north of us, and that's this small island of Angar, one of about a dozen or so inhabited islands. There's over 700 islands in the Palauan Islands. Angar is one of the smaller ones. There's only about 120 people living on it. Historically quite significant. It was was also the stage of some significant battles in World War II. And the place where we're diving here, we're just outside just inside the boundaries of the Palau National Marine Sanctuary. So the boundaries would be about 5 miles here to our north. And the sanctuary extends from 24 nautical miles to the end of the exclusive economic zone. For most of the area around Palau, with the one exception currently in the northwestern quadrant, there is about 20% of the EEZ that is part of a domestic fishing zone. The other 80% are part of the Palau National Marine Sanctuary, which is one of the largest marine protected areas in the world.
Yeah, I don't see it.
And here we see a very good example of a lot of those colorful corals that you saw earlier. They all need rocky outcrops to grow on. All this rubble, soft substrate is not favorable habitat for them. And so that's why you don't see those here.
So, question from our viewers. Has there ever been anything that you've seen with a herc that has given you the heebie-jeebies?
Those sea spiders.
Well, last night in the lab, actually, there was an associate with that Phrygian sponge that gave a few of us the heebie-jeebies. It was a very long polychaete that took a while to pull out of the sponge. And our other science manager in training, Noel, was very spooked by it, and we had to we all had to trade off pulling this out. I think being really up close to some of these amazing organisms can be moments of total awe and sometimes moments of heebie-jeebie.
Yeah, and Ayla, Noel, and May have been doing an exceptional job working long hours after the ROV comes on deck. Basically taking the samples from the ROV, bringing them into the lab, photographing, documenting them, preserving them, labeling them up until long hours after the dive has passed, and that's just the beginning. After that, they also type up reports, summaries of every one of these dives, and a lot of data processing. So I really appreciate, because that's really enabling a lot of the follow-on discovery that will happen for these expeditions, is to have properly documented samples and summaries from these explorations to allow researchers to use the data that we collect. That is a big part of our mission, is to collect as much information as we can with these explorations, but then make it available to people from around the world so that it can fuel additional science and activities. And so yeah, that work that happens both during and after dives to properly archive the data we collect is very important. And also a big shout out to our data engineer, Michael Labriola, who is also an absolutely critical component of that workflow. There's just enormous amounts of data that are collected on every one of these data.
Can we zoom on the white coral?
All right, push in there.
Alright. I think this is a Paraclyptrophora. It's got lots of associates. Thanks.
Okay.
It just helps us confirm what I thought it was.
Nice. Come back, ahead.
What's the difference between the Calyptrophora and Paracalyptrophora, Megadita?
So the Calyptrophoras tend to be very lyrate in the way their branches are spread out. The Paracalyptrophora
We're going to do a little pilot swap here.
are less lyrate. They have a little bit more jumbling in the way the branches branch, but so they can look very similar in some ways, but also the branching pattern is very distinctively different.
Copy that. Thanks, Megan.
You're in auto heading and stick lock.
So Goose and Maverick are swapping here.
Which one's which?
Well, I'd have to give you my mustache, I think. So you can see the slope is like perpendicular to the way we've been traveling. So I've been like crabbing along, looking at the slope as we go. Yeah, yeah. Yeah, the ship hasn't started moving yet, so.
So we have a question. What kind of bacteria is found in the deep sea, and are there any sea, deep sea fungus or mold.
So yes, absolutely. There's bacteria, and then most abundant and most diverse here. I am not a microbiologist, but yeah, that's a active area of research. So there's also a lot of specialized type of bacteria that grow on certain environments, including on certain tissues of the different organisms. And there's, yeah, a whole bunch of researchers that are trying to just sequence everything that lives on these organisms. Obviously, in these types of explorations, we're mostly focused on what we can see with our naked eye and with the cameras of the ROV and what we can sample. And among the things we can sample are the things that live on the things we sample. And yeah, all of the things we've been sampling, including the rocks, every single one of these animals is covered with bacteria. So they could potentially help support studies as well of the bacterial organisms associated with certain environments.
There's also a lot of bacteria known to thrive at hydrothermal vent ecosystems. So not like these ones that we're seeing here, but ones that are at volcanically active regions where the seawater goes in the crust, heats up, comes back out like a hot spring, and bacteria is kind of the primary producer in the sense that it gets its energy from those hot fluids, and they can come in so many different morphologies. Some look like fluffy flowing cloaks over the towers, and many different kinds of bacteria live in the deep sea at whale falls as well. And as Daniel said, on organisms in the sediment, they're very abundant and mostly, I would say, uncharacterized. There's tons of bacteria species we don't know about.
Nice. Coming up on the winch. Your mic, yep, a little closer. Good still cam shot. Nice.
So we have another question. When planning a dive, are there a specific set of variables that you use to determine what route you will explore across the seabed?
Yeah, absolutely. We look at a whole bunch of things. So, we start off by, what's the basic scientific objective we're trying to accomplish? Are we trying to go across a variety of different habitats? Are there a specific habitat that we're after? Are we just trying to cover gaps in previous surveys? So that's kind of the first set of parameters that we look at. Another important thing that we look at is safety of the vehicle. So we look at where we expect the current to be in, what kind of the currents are going to do. And so we'll approach features based on what would be the safest approach for the vehicle. So if you had some kind of sudden failure, you would want to drift away from certain cliffs or things like that, especially if you consider that you have this long tether above you. And then you just also study the maps, and the maps will give you kind of clues. A lot of the organisms, a lot of the geology that we're after are kind of after places where we expect, we see kind of interesting topography, where you see a bit more texture in the maps. And then we plot these tracks, basically doing that balance of, one, we want to achieve the scientific objectives, whatever is set out to. Two, being safe as possible for the vehicles. And yeah, it's a combination of factors. And the one thing is, about this type of research and exploration, it's different than what many other researchers do, is where you have a very specific objective. We are not after just one thing in these explorations. We're basically we don't really know what we're going to find in these environments. So it's a little bit more open-ended when we design these surveys, and we're basically trying to cover as much different types of habitats. And that doesn't just go for this particular dive track, but it also goes for the expedition as a whole. So if you look at the geomorphology of Palau, it's quite diverse, from shallow water reefs to the Palauan Trench, submarine canyons, seamounts, plateaus, ridges. There's all kinds of different features. And some of those features are too deep for us to study, so we are limited on this expedition to diving to depths of 4,000 meters. And so we basically try to look at a cross-section of different habitats within those top 4,000 meters that are within the Palau National Marine Sanctuary, and also use previous data, previous surveys. There's been a few dozen surveys around here with different vehicles, and so
are you trying to land, or
using ours, layer those on top, so that we're not replicating previous surveys, but building on those previous surveys and starting to basically fill in those big gaps.
You want to turn the other way?
very big colonies here.
So this is a different species of anemone in the family Actinostolidae. It's got these really thick tentacles. Oh, the wacky waving inflatable arm flailing tube men.
So just as a time check, we have about 8 hours, a little over 8 hours of bottom time to go on this dive, and so about midway through this dive, and we are approaching our midway point in the track, so hopefully we can cover some ground here. This section here between waypoint 2 and waypoint 3, for those who are following on the dive plan, is a bit flatter. Once we approach waypoint 3, it'll start getting a lot more inclined, and that's going to be a steep ascent to a topographical high point along this ridge. And there's about 3 or so high points, basically tiny mounds on top of this ridge, and we'll try to get to at least one of those.
That's that same fish again. Yep. It likes to taunt you. See, it's even opening its mouth. Nope.
Also great to see some Some new faces and new names here in our scientist ashore community. We got Kurt Bryant Bacharo from University of Hercules. Welcome. Thanks so much for joining us.
Cool shot on Atalanta. Atalanta.
Keeping our eyes peeled here for any organisms that we might have not yet spotted throughout the dive. It looks like we see a lot of common. Definitely this yellow acanthogorgets being very dominant. Yeah, let's take a quick look at, right at lasers. I think a Brazingid. I don't recall seeing them earlier.
I think it's a crinoid.
Let's see, yeah, could well be. And I think you're right, yeah. Yep.
Yep, and there's also some hydroids on some dead coral and acanthogorgia and a paragorgia and an Enelopsamia. There's also some small ferriid sponges and a bamboo coral. Some other hexactinellids or glass sponges, little ones.
That's great, thank you. Yeah, another one of those very large chrysogorgid sea fans.
Yeah.
Heterocarpus shrimp.
Atalanta's getting ahead of you.
Yeah, amazing how this rubble, yeah, how extensive.
This looks like it could be a rock pen.
Yep.
So rock pens are a type of sea pen. And the rock pens have a specialized base. It's kind of got a little suction cup. That suction cup helps it adhere to the seafloor. So this looks like an anthoptylum, a type of sea pen. That's like pretty average size for them, but they can get a little bit bigger.
Yeah, it's great that we're covering ground.
Did the photogrammetry stop? Yes. Okay. You turned it off. Okay. When around when?
Look at your nav screen.
Just
come,
so
like,
I can
back
note
up
it.
slope a bit. To the.
Like a half hour ago. Yeah. When we started going upslope. Yeah. Okay. Okay. Yeah. Yeah. No worries. That's totally fine. But just have her end.
So Daniel, one of the tertiary objectives of the expedition was to look at maritime heritage sites, either cultural locally or something in the more modern era. In this particular area, were there any particular wreck sites that were known to go down but unknown what their locations were that might be something that could be popping up in the expedition?
Yeah, so great question. So when we first set out to plan this expedition, and maritime heritage is always something that a lot of folks are very interested in. When we talk to a lot of stakeholders here in Palau at Historic Preservation Division, and yeah, there was a lot of interest in stone money. So there's been trade routes between Yap and Palau going back long time ago, and a lot of those large pieces of stone money are known to have been lost, and that was a big interest to make that part of our search. Unfortunately, with our mapping systems, we would not be able to detect something like that. Something would really have to be over 100 meters or so for it to be picked up with our sonars. So there is obviously a potential that we might be able to pick up something like that, but if we get very lucky and on an ROV track go over it. So that was one interest, and then the remainder of the specific sites that we got were all World War II wrecks. So in 1944, a whole bunch of ships were sunk around Palau, and yeah, they range in size, and they're all either Japanese or U.S. In terms of probably some of the more known ones of those. From the USS is the submarine, the USS Tullaby. So that was a very known, and that's a submarine about 100 meters or so long that suffered the ill fate of having sunk itself. A torpedo that basically it shot and circled around and hit itself. So that was one site that we have coordinates. The last known position is in the northern part of the archipelago, where we had several days of operations, and we tried to map around it and didn't detect anything. And then there was many others. We had to narrow the list. We had over 100 or so wrecks that are known within the Palau National Marine Sanctuary, and I should not say known, they said the last known position is in the Palau National Marine Sanctuary. We of course don't know how accurate those positions are. In some of those cases, it's a radio position from a different vessel. You have to consider that these positions might at some point, someone is writing something while they're at war, and it's also obviously technology back then was very different. And so yeah, those, you have to take them with a lot of grain of salt. There is a couple bigger ones that we were trying to locate, including one that we did some mapping actually last couple days, the Boston Maru. That was a large cargo ship, and it's somewhere in the eastern corner of the Palau National Marine Sanctuary, lost, and 246 people lost their life during that. Sunk by a torpedo. That was one of the bigger ones. Yeah, I mean, the list goes on. There's just very many. The San Clemente Maru is another big tanker ship that was also sunk within it. And I mean, I could read the list here, but they're all different sizes, different, and we narrowed down the list from those 111 to about 15 or so that are bigger ones, and that we actually thought we would have a reasonable chance. We'll continue mapping throughout these next couple weeks when we're out on the next expedition, and hopefully we might be able to detect something with our sonars, and especially in the backscatter, we might be able to detect something and then plan a dive. And yeah, we'll see. It's it's definitely looking like looking for a needle in a haystack, but it's always very interesting. And now there's a long history of marine conservation areas being designated or promoted because of cultural heritage. So in the United States, we have several national marine sanctuaries built around cultural heritage sites, both in the Channel Islands and also in the Great Lakes, also actually in the Florida Keys. And I think these underwater cultural heritage sites, they have, now obviously a lot of the focus of these protected areas is on biodiversity conservation, but there's also human history conservation in these places. And I think one important element is that they help connect people and global audiences to these places, and that helps perpetuate and provide awareness about these places. And if these places are out of sight, they tend to become out of mind. And these stories, the history of these things, they really have an important role in conservation. And so, yeah, that is why we were also trying to make as you said, a tertiary objective of these expeditions was to try to find it, because we've seen that these things can do a lot of good for conservation areas. They create a lot of awareness, create a lot of connection with different people from different parts of the world. And so, yeah, we'll see. We'll keep looking, and maybe we'll find something.
Are there cases where you collect artifacts from these shipwrecks or aircrafts and are later brought to museums for display?
Yeah, great question. So first and foremost, we only do activities when we have the permits to do so. In general, our approach is look but don't touch, look but don't disturb, leave nothing behind. And so our general approach is just to document with videos how that's important, and some of these are fragile, and in some cases these are grave sites, and that's really important to realize. So we want to approach these areas with respect and with a lot of dignity, and that's these are sensitive things. And so in the past, so the Ocean Exploration Trust and EV Nautilus has been around the world and has, over the years, documented and discovered close to 100 shipwrecks around the world. Work in many different places, including in the Black Sea and the Mediterranean. In some of those cases, we did have permits to recover an artifact to basically enable then the follow-on studies of trying to understand what these artifacts are. But our first general approach is basically do what we have permits, and in most cases we just ask for permits to just take photographic or video graphic evidence of these places, and then make that information available to management authorities, who then could do follow-on studies. And has all kinds of different management implications. I talked about the importance of wrecks, like connecting people to these places, raising awareness. In some cases, some of these wrecks are potentially polluting wrecks. So some of these wrecks might have gone down with lots of oil, or and then these have been down in the sea for a very long time, and some of the metals over time corrode and it could release oil. So that's a hot topic in a lot of places, potentially polluting wrecks. And so, trying to locate them is then also the first steps to try to do a remediation. And so it's one of many objectives that we try to weave into these expeditions because it has both scientific and management implications.
Another thing that can be helpful with characterizing wrecks or just all of these sites is using those imagery and video for making 3D models through photogrammetric methods. This can be a way to visualize those 3D features and historical sites and structures in a way that can allow for a different kind of analysis that is maybe a bit more similar to what you would get with actually holding or looking at something in 3D. So I know there has been previous wrecks, whether it's airplanes or ships, that use that technique, not just to document it, but also to learn a little bit more about how maybe that that wreck happened, what was the cause of the sinking. So there's a lot of non-invasive methods to document and learn from sites like that and have some closure for those who are lost.
Yeah, I think thanks for mentioning it, May. And then, that was as you alluded to, those are powerful tools for science because they allow you to explore the wreck after you've removed the ROVs, and you can still kind of almost virtually dive on the wreck and look at it and spatial features. But they're also powerful tools for education and outreach. There's actually the Bureau of Ocean Energy Management has a virtual museum of shipwrecks, basically where they've imaged a whole bunch of these, and you can explore them at your own leisure.
Another great resource if you're interested in wrecks, especially in Hawaii, there is the Hawaii Undersea Research Laboratory Archive website. You can just Google HURL, H-U-R-L, archive, and that'll bring you to the website. And there is a maritime heritage section that I helped assemble, and it has a bunch of photos and information about the various wrecks that have been found off the coast of Oahu, including submarines, ships, airplanes, automobiles, and amphibious vehicles.
Megan, how close are we to waypoint 3?
We're about 50 meters.
Oh, wonderful. Yeah, that will be a perfect handover to our incoming watch. We'll have the carry on the baton. That'll be kind of the midway point of the dive track. I'm also a little sad to say that this is going to be the last watch that I get to share with this team on this expedition. It's been absolutely wonderful to share the 12 to 4 with all of you. It's been a really remarkable team here.
Are you not staying for the next?
Yeah, I'll be on, but we'll have a couple new faces, so this constellation won't be the same, but
Okay.
but yeah, it's absolutely wonderful. Our human encyclopedia Megan, are absolutely superb. Maverick and Goose, the ROV pilots that just with surgical precision, Sarah and Jake. Also just gained a tremendous appreciation for the work that Aaron does, our video engineer, as we've been struggling with focusing a camera here in the back and knowing how much work that is and how how much you have to concentrate. So thank you, Aaron, for bringing us those stunning images.
Thanks.
Yeah, May also learned so much from you, and yeah, every night after the dives are over, I review the notes and review the data logging that happens, and it's just such a powerful way to go through a dive and remember what some of the key moments are. Makani, it's been awesome to have you here and learn about the work that happens at Palau International Coral Reef Center. And similarly, Imade, yeah, just really, I know we've worked together in the past, but it's awesome to have been shared these watch together.
Yeah.
And obviously, Jeremy, the first time we get to sail together, such a privilege and pleasure.
Yeah, thanks, Daniel. Thanks, everybody, for letting me sit in. This will be my last watch sitting in, too. I think John Culberson might be popping in here afterwards. I don't want to be hogging up this chair, but I haven't been back out to sea since 2016.
Oh, wow.
So it's great to
Yeah, the current's pretty strong.
head back out here. So thanks for letting me join y'all.
Thanks for joining us. We love having you here.
Oh, careful, because I might just leave DC permanently and come back out.
And thanks for the kind words.
Yes, very kind words, Daniel.
Yeah, thank you.
Yeah,
You're the best.
thank you for the leadership and all the wonderful comments and contacts. It's been great.
Okay, so our viewer is asking, why is there a lot of coral rubble in this area?
Yeah, let's see that sea star.
Yeah, we'll focus in on a sea star, and then yeah, we'll take that.
It's a little deep sea ravioli.
Is it?
It looks like a ravioli.
Oh,
I don't know if it's the ravioli.
the ravioli. I don't know if it's the ravioli. It looks a little bit too cookie-like to me.
Yeah, well, this is definitely a cookie star.
Yeah.
So this is a sea star in the family Goniasteriddae. Oh, now that we're getting a good zoom of it, I can consult the guide. It might actually be in there.
Okay, that definitely looks like a pastry.
It does, right? I could definitely go for a cookie that looks like that.
I was going to say, is everyone just hungry for dinner?
Cookies.
So it could be something like pleauphaster. Yeah, that's going to be my best guess, is pleauphaster.
I think Jeff wants us to collect it.
We have to stop the ship.
Sure.
Yeah, if we're set up to do so, we should go ahead. Yeah,
Bridge
thanks
nav.
for catching it, Mike Honey.
Do you want me to pull up? Yep.
I'll do this one.
Can we hold position?
Maybe.
Are we slurping?
It's a good question. I don't know.
Yeah, I think we should suck it.
I think it'll reach, yeah. Nice.
Yeah, no, it's not going to run away like all the other things that we've been trying to suction. Yeah, I think you might need to scoot.
Yeah. Well, I don't know.
You try hard enough.
You can bump it all the way up. I'm afraid if we suction too low, it's not going to go all the way through. So. Might be too big. Yeah, I think it's going to have to be put in the forward box. It's too big for the slurp.
Roger.
You bum with the box?
If we're confident we're not going to use
other
the
box.
scoop, maybe we'll put it in that box. Is that,
Okay.
yeah. So
Yeah, I think that's a good
let's
idea.
go for the starboard side.
Now you have to write that big note in all caps that says,
Forward.
"Do not use scoop."
Oh, yeah?
You have to write the note in all caps like they had for
Yes.
you that says, "Do not use scoop."
Exactly.
That's all right.
It might try to run away.
A puff pastry.
All right. I think it should drop in there.
Nice.
Ooh, good job, Sarah.
I see
Congrats.
you didn't use the grabber directly this time, Jake.
Oh no, you should have seen me earlier though.
Huh?
I redeemed myself.
Did you?
Yes.
Well done. Great sample. Thank you. Hey, Nav, could you drop a waypoint for that sample? It's 104.
Doing what?
Committing ROV crimes.
Oh, here comes Dan.
Coming up on the winch.
It's my fault. It was getting too hot. The maximums that we're given for the computers.
Good afternoon, everyone. And a fine one it is. Look at all the sclerotinian rubble. Question is, are we gonna find where it came from?
Stay tuned to find out.
And Brian, tell us what that is.
Yeah, so I'm looking at the sediment, the sea floor here. It's not really sediment. Well, there's sediment too, but this is basically all dead, hard coral skeleton. And it's kind of multicolored because it's different ages. The really black stuff is fossilized and has been encoded with manganese crust. And then the lighter colored stuff is more recent. Really pretty Iridogorgia there going by. And Jeff and I were just looking up some papers on this in the lounge about where the aragonite saturation horizon is, and it's projected to be about 100 meters deeper than here, which is about the same time we started seeing the Inelapsamia and the Madrepora start showing up. But this level of density of coral skeleton is impressive. So we might get a little higher and see a really significant live ring, but also this could be thousands of years old. And so we may not see a lot of live sclerotinian up here. So we will only by pushing on what we know.
If you turn to your right as I come by Jaden, we'll go up the hill and get Herc on the shallow water side there, so you're not scraping the seabed.
Copy.
So how would you all feel about pulling the same maneuver we had yesterday and sliding over into the steeper stuff on the east side of the feature and then jogging up, doing another 3.5?
I'm following my nose up
right around here?
Yep.
All right.
I mean, basically this looks like Groundhog Day in terms of high pack.
Every day is just like Groundhog Day.
Well, every day is a Tuesday, but...
That it is.
Are we okay to start with a couple introductions? All right.
You ready to get the ship moving, Dan?
I need to know what direction you plan on moving first.
065.
Okay.
Yeah?
Yeah.
Let's do it. Bridge nav, 50 meters, 065.
I get uphill this way.
Yeah, where we're going.
I mean, immediate uphill, local uphill.
Local uphill.
Northeast, looks like.
If you want to jog that way and do a little move up, that's fine with me too.
It's not that steep. There was a little.
I mean, my goal is just to get on the other side of the ridge and see how the other side is facing the different current might be different.
All right.
However you all want to do that, I'm open. So, introductions, yes, right. As we're doing next?
Yes.
Cool. Hi everyone, I'm Brian. I'm the watch lead. I'm a deep sea benthic ecologist. And when I'm on shore, I am a visiting researcher at Boston University and the chief scientist at the Ocean Discovery League and the external science advisor for NOAA's Office of Ocean Exploration's Pacific Islands campaign.
Ali everyone, I'm Harold Carlson. I'm a PhD student in oceanography at the University of Hawaii at Manoa. I'm also a deep sea ecologist, and I'm in the science chair for the 4 to 8.
Ali, and hello everyone. I am Ayla, and I work at the Coral Reef Research Foundation as a Marine Lakes Research Assistant, and I'll be the data logger for the 4 to 8 watch.
Hey, I'm Chris Krasnowski. I'm a full-time contractor in ocean robotics and mapping, and I'm here today as navigator and high-resolution mapper.
Dan in the Herc chair, general roustabout and all-around troublemaker.
Hi, I'm Jaden. I'm the Atalantico pilot for the 4 to 8 watch. outside of Nautilus, I'm a full-time mechanical engineer at the University of South Florida.
And hello, I'm Amber. I'm the video engineer, and on shore, I'm a cinematographer and camera operator based in Los Angeles.
And I'm former Congressman John Culberson from Houston, Texas, a longtime supporter and zealous advocate for telepresence and ocean exploration and the work that Dr. Robert Ballard and his team here on the Nautilus have done. And this is my 8th Nautilus expedition that I've been a part of. It is an extraordinary experience with a great team of people who are all dedicated to the mission. And we are mapping and exploring parts of the world that have never been seen before, identifying new species, new geologic features, and new sources of mineral wealth, and looking to identify lost World War II vessels as well. But it's a privilege to be here with all of you and to have worked with you all these years in Congress.
Yeah, thank you for all of your support and advocacy.
And last but not least, my name is Elle, and I'm in the Science Communications Chair. Onshore, offshore, when I'm not on the EV Nautilus, I am a researcher at the Palau Community College under the Cooperative Research and Extension Department. And this is your 4 to 8 watch team. So a little bit for our viewers on where we're at and what we're doing. We are at an unnamed ridge south of Angar, roughly 30 nautical miles southeast, and just inside the boundaries of the Palau National Marine Sanctuary. There have been no previous dives that have been conducted at this site, although there have been several previous dives using the JAMSTEC manned submersible Shinkai 6500, which surveyed depths to below 6,000 meters, which is located southeast of this ridge. This dive plan is to survey in unexplored habitats that are shallower and those explored shallower than those explored by JAMSTEC previously. So we are at our maximum depth was 1,158 meters, and we are currently at 774 meters.
Which I believe is the shallowest we have explored in Palau so far. So this is new depth for us.
And are we fairly close to the island of Peleliu, where that historic and awful battle took place in World War II?
We are close, but it's closer to Angar, yeah, the two southern island states of Palau, Angar and Peleliu.
But we're right on the border between the artisanal fishing zone and the start of the Palau National Marine Sanctuary. And geologically, we're just west of the Palau Trench.
And Brian, for those folks just joining us, what type of trench is the Palau Trench? You know, there's spreading zones, there's subduction zones, and what does that mean for people that are listening?
So this is a subduction zone, and so this is where the Caroline Plate, which is a minor plate, part of the Pacific Plate, is being subducted under the Philippine Basin Plate, which is also a minor plate of the Eurasian Plate, tectonic plate. So this area is really tectonically super complicated. And so it's a very, very slow subduction zone. So there's some earthquake activity, and for those of you who live on the east coast of the United States, like I do, there's a lot of volcanic activity, but compared to, say, Japan, there's not as much here. And there isn't any active volcanism or anything we know of here, like you would get like in a back island arc like you have in the Mariana Islands. But this is still a subduction zone that is active, and it's just a very, very slow one. And we are just, basically, if you turn around and continue in the direction we're going or go a little more east than we're currently going, you would jump over the top of this ridge, which we're about to do here shortly, and then it would continue to go down into the hadal zone below 6,000 meters. Actually, I guess it's the abyssal. Doesn't quite get to hadal depths here.
So what we're looking at right now, it looks pretty different from our previous dives. And actually, to me, it looks like seagrass a little bit.
Yeah, it is pretty different, and I'm pretty interested. We saw the start of a little bit of this yesterday at a little bit deeper depth, but a lot of this is old sclerotinian coral. So those are actual, like, reef-building corals like you would see in the shallow water scuba diving around Palau. And they are generally only found in this part of the world shallower than about 1,000 meters, which is about where we picked them up here. And what I'm going to be really intrigued by, in some places these are just found in little pockets. Like yesterday we saw a fair number of different types of reef-building corals or colonial sclerotinians. But we didn't see widespread skeleton like we're seeing here. And so this could be relic from a previous geologic era where we had different climate and different oceanographic conditions that were more favorable for this stuff growing here. And we just see a little bit of live coral left. Or we might just be downhill of a larger aggregation of it, and this is just being moved downhill. And we won't know until we get a little bit higher up this feature.
Brian, how old could these old crumbled up dead fossilized corals be?
So a lot of that depends on the paleoceanography here and how the aragonite saturation horizon has changed over the course of different climate periods. But tens of thousands of years old would not surprise me at all.
Care to explain what the aragonite saturation horizon is?
Nah, why don't you do it?
Oh, I had to do it last time. Okay, fine. So, basically, animals in the ocean that build calcium carbonate skeletons can only do so under certain chemical conditions where the right building blocks are available and you have the right pH and the right temperature to allow those chemical reactions to proceed forward. And below a certain depth in the ocean called the calcium carbonate saturation horizon, those reactions are no longer favorable.